So good morning. Has this been a great conference or what? I want to thank everyone, all of the patients and also all the physicians that spent time with us. And I've had a ball. It's been really awesome personally, meeting as many people as I can and spreading the word of education for Waldenstroms. So for anyone that's been in the community for a while, there's really no introductions needed for Dr. Treon. He's very well known to all of us. But for the newcomers, a little background on him. He has been a special friend to the Waldenstroms community for years, and he's really been an advocate for educating patients and physicians. And to that end, he is the director of the Bing Center for Waldenstrom's Research at the Dana-Farber Cancer Institute in Boston. And he has arranged many times to come out to the patient support groups to speak to us as Waldenstrom's patients to educate us about our disease and the research that's being done with regard to finding a cure for us. So we have to thank him for that. He's also been instrumental in educating and bringing together academia and the physicians and researchers that are doing research to the cause of our disease. And to that end, he's had organized the 6th International Workshop in Waldenstroms in October of 2010, which was in Venice, Italy. And he's done the 4th International Patient-Physician Summit in Orlando this past March. I am personally thrilled to be able to introduce him today because I've been the liaison for the IWMF for a grant that has been supportive of the work that he's doing at the Dana-Farber. The title of his talk today is The Comprehensive Studies into the Genomic Basis and Pathogenesis of Waldenstrom's Macroglobulinemia. So please welcome Dr. Steve Treon.
Thank you very much, Ron, for that very kind introduction, and to all the members of the board for inviting me here today to be able to share with you the progress that's ongoing in this disease. And for starters, I do have to mention that there's a lot of new data that I'll be presenting to you, including data that I've not had the opportunity to publicly present before. But being among family members, I think it's important for you to be able to see this data firsthand. So let's talk a little bit about Waldenstrom's. So one of the areas of interest that we've had in particular has been to try to understand the genetics of this disease, as was alluded by Ron earlier. And one of the angles that we've taken in terms of understanding the basic science behind this disease is to study in particular those patients that have a familial predisposition. And there are a lot of examples in cancer genetics where the discovery of the genes that actually emerged that define those cancers actually came from family studies, such as the one that I'm going to talk to you about. So this is actually data that we just presented at the International Conference on Malignant Lymphoma in Switzerland, which looked at the familial predisposition in Waldenstrom's, and it involved a huge series of patients at our institution, over 1,000, as you can see, consecutive patients. And what we defined in this patient population is that 26% of the patients actually had a first or second degree relative who had another B-cell type of cancer. And as you can see the distribution here, when you look at the relatives of the individuals that have the Waldenstrom's, the most common other lymphoma that you see is actually that of other non-Hodgkin's lymphoma types. And then you see CLL, chronic lymphocytic leukemia, and then you see Waldenstrom's running in those families, as well as multiple myeloma. And so it gives you a little bit of a breadth that patients who have this familial version of Waldenstrom's may be predisposed to a gene that actually may lead to all kinds of other lymphomas. Now, as Ron alluded to, one of the great advantages given to us in this study was a large grant by the IWMF that allowed us to set up the Waldenstrom's Familial Genomics Project. And this involved bringing together families of patients with Waldenstrom's, both those that had familial disease as well as those that had sporadic. Sporadic meaning no history of any type of Waldenstrom's or other lymphoma in their families. And this accrued over 800 individuals, 187 families to whom we collected very detailed family history, but we also collected lots of samples. We analyzed their blood cells, and we analyzed their immunoglobulins, and we also collected their DNA. And at the end of the day, what we discovered was there's really three types of patterns that we could surmise. One is, of course, the sporadic pattern, which means just an isolated Waldenstrom's case. The other is where we see only Waldenstrom's running in the family. And this is a pedigree where the grandfather had Waldenstrom's, the son had Waldenstrom's, and both of his children have Waldenstrom's. And then what you see is the so-called mixed B-cell category where patients come in with all kinds of history in their family. This is a grandmother that had CLL, her son had Waldenstrom's, and his son had non-Hodgkin's lymphoma. And on the other side, which is quite interesting, there was a skip in the generation here. There's no disease detected here, but the grandson of that patient had non-Hodgkin's lymphoma. So that implies that even though the predisposition may be there, it doesn't always get enacted on. So there may be other factors that play an important role. Now, how does the 26% figure in terms of familial predisposition stack up to other lymphomas? Well, this is an interesting study from Jennifer Brown at our institution, where she did a prospective analysis of almost 2,000 patients with B-cell lymphomas. And as you can see, looking at first and second degree relatives, among patients that had CLL, only 17% actually had a first or second degree relative with another type of B-cell cancer. Among the non-Hodgkin's lymphomas, it was 12%. Among the Hodgkin's lymphomas, it was 11%. So, you know, the relative number of 26 is pretty high when you stack it up against another very large database. So one of the interesting questions that we've had, and I know Mary McMaster has been here at the meeting, who has been really a leader in understanding the familial dynamics of this disease, but one of the unanswered questions that we've had in our field has been to try to understand, does this really mean anything if you have familial predisposition? Well, of course, it could mean something if it involves your children and you're interested in knowing what their risk is, but it's also important to know if it has an impact on the prognosis and treatment of the individual. So this is a study that we just presented in Switzerland at the International Conference on Malignant Lymphoma, where we actually looked at the impact of familial disease in patients who had never received Rituxan and then went on and received a Rituxan-containing regimen. There were 135 patients represented, and roughly a quarter of these patients had familial disease, and their disease characteristics were quite balanced between the two groups. And what we found was that, in fact, having familial disease did have an impact on how well those patients did with therapy. In fact, as you can see here, whether we look at the overall response rate, the major response rate, meaning that at least 50% of the disease went away, or the attainment of a complete remission or a very good partial remission, which means at least 90% reduction in disease burden, patients who had sporadic disease did better than those patients that had familial disease. Now, when you also look at the impact of familial disease on how these patients did in terms of the time that their disease could be kept in remission or the time that they could actually avoid going on to another therapy, once again, the familial patients did worse. Now, it's one thing to be able to tell somebody bad news, but it's good also to be able to tell them some good news. And so when we went back and did a subset analysis of the patients, we wanted to see if the type of treatment for these familial patients actually made a difference. And what we were surprised with, even though these numbers are relatively small, is that the patients that received Bortezomib-based therapy or Velcade-based therapy actually did better than those patients with familial disease that did not receive a Bortezomib-containing regimen. In fact, you can just see these bars right across here, whether it's the overall response rate, the major response rate, or even the attainment of those very good partial remissions or complete response. Look at the difference here. Only 6% of those individuals that got a non-Bortezomib therapy got into that very good partial response category, whereas 80% who got Bortezomib did. So, the findings of the study would say that if you've got familial disease, maybe you're better off, with a Bortezomib-containing regimen. And those are the kind of studies that need to be done. And this is also an interesting slide because it also shows you, the progression of the disease in those patients that got Bortezomib-based therapy versus non-Bortezomib. And if you were familial, you could actually get a better and longer remission with Bortezomib compared to non-Bortezomib therapy, whereas the patients that had sporadic disease, it really didn't make a difference. They both had the same time that they were kept in remission. Now, in looking at the therapeutic options in this disease, I think many of you will appreciate right off the bat that rituximab has probably played the greatest role in our ability to get patients into a remission with Waldenstrom's. And I was particularly privileged to have had the opportunity to have published the first report of a response in an individual with Waldenstrom's who had relapsed disease and received rituximab. And many of you know about the molecule. You know that it's partly mouse, and therefore there's Mickey for you. And it's mostly human. And so this is an important consideration because when these antibodies were being developed, it was important to humanize them so that they could interact with human immune cells. Now, one of the things that we discovered along the way about rituximab was that it had some interesting quirks. And one of the quirks that I'm sure you've heard about at this meeting is that it can induce your IgM sometimes to go up very abruptly. We now have a term for that. We call it the IgM flare. And this was one of the earliest reports of that from our institution, in fact, showing how these patients ended up flaring. This is actually somebody that I was caring for, where her IgM was 7,500 before she got the Rituxan, and it went up to almost 12,000 4 weeks later. And her disease burden didn't change one iota, and she ended up in the ICU with a major head bleed because her viscosity level went up abruptly as the IgM went up. And so that was a lesson to us, that for patients who have, high IgM levels, we either plasmapherese them or we try to avoid giving them Rituxan, at least initially, until the IgM level comes down to a safe level. But it also taught us another lesson, which was many times we would get calls from physicians saying, well, the IgM is going up, the patient got Rituxan, I'm going to switch chemotherapy. And that was also an error too, because many times those patients are actually responding even though their IgM is going up. Now, why does this happen? This has been the subject of work in our laboratory for just about a decade now, because this was of great interest to us. And what we discovered along the way was that IVIG, which sometimes patients get because their normal antibodies are low, also cause this IgM flare to happen. So if you're going to be getting IVIG at any point, it's important also to make sure that your IgM levels are in safe range. But it also told us that maybe it wasn't the Rituxan binding to the CD20, the target, that was causing this flare, and maybe it had to do with some common feature of both antibodies, which was the other part, the Fc part of the molecule. And so Guang Yang in our group did very nice studies where he actually looked at other immune cells cooperating in this flare process. And he did these co-culture experiments where he took Waldenstrom cells, added in other immune cells, but in particular the cell called the monocyte, and then added in the Rituxan, and lo and behold, the flare happened in the test tube. And so he went on very nicely to work out the mechanism and show, in fact, that these monocytes release large amounts of IL-6. And just as Steve Ansel told you, IL-6 is a big stimulant of IgM production by Waldenstrom cells. And if you look at the patients that are flaring, their IL-6 levels go up, before the flare happens. Now, Rituxan has clearly been an important drug for this disease, and in fact, whether you get 4 weekly infusions or 8 weekly infusions, you do see responses, but the responses are usually in the level of about 25 to 40% of the patients. And almost never do you see a complete remission.